It's a pleasure to introduce Trevor Pitcher from the University of Windsor. Trevor is the acting director of the Great Lakes Institute for Environmental Research at Windsor. He is remarkably adept at getting funding for infrastructure. He runs a fish hatchery, which he uses for his research. Trevor has focused on fish ecology, evolutionary ecology, and has developed a career integrating evolutionary ecology with conservation in a way that I've admired for quite some time, and I'm very excited to have him here talking about his work. And he's, just as an aside, he's got 2 students in the Renew Zoo program, which is yeah, we'll talk about that later. All right, thanks.
Great. Thanks for a great introduction. And frankly, it's a great privilege to speak after our first 2 speakers because they framed things so well. The only downside of going after them is they're going to make me look really bad. But the main point, as Albrecht mentioned, is that I do a little bit different. I work on fish species primarily, although amphibians have come into my realm recently. And what's interesting and different about fish in terms of reintroduction, as you might think about, is that a lot of fish species have a large number of gametes and offspring. So that is to say, unlike many mammal systems that have a few individual offspring over their lifetime, some fish, including sturgeon I work on, have millions of offspring and eggs within their lifetime. So the capacity to deal with reintroduction often is beyond the zoos and aquariums. And so what I do is I try to find that interface between both aquariums and zoos and also the provincial and sometimes federal hatchery system, if I'm in the United States, to work on these reintroduction programs. So my talk today will be more focused on the biology of reintroduction programs, and specifically talking about restoring aquatic biodiversity in the Anthropocene. And the Anthropocene, as Dan mentioned earlier, really refers to the idea of this new era that essentially is human-induced changes that often negatively impact species. And the second part you can see is developing the science of reintroduction biology. We heard both Dan and our speaker before later talk about these ideas of reintroduction, but reintroduction biology is actually an incredibly young science. The first 2 formal textbooks on reintroduction biology only appeared in the last 2 years. So if you think about that, it's an incredibly naive science that has a long ways to go. And one of my goals within my career and with many colleagues is to better to develop these ideas. And one of the things Albrecht mentions is to infuse ecology and evolution into this process of either captive breeding or reintroduction biology. And although I primarily focus, as I said, on aquatic species, we work on a variety of systems, including the reintroduction to Lake Ontario of Atlantic salmon, which used to be historically abundant. And for Dan's sake, it used to be Samuel Wilmot in the early 1900s who tried to resave them after European colonization took them down to very low levels. We also work on lake sturgeon, shown in the top right, in the Maumee River, and that's a collaboration I'll talk about later with the Toledo Zoo. On the bottom right is red-side dace, which is provincially listed and soon to be federally listed again. And we'll talk about what's being done for their work. And then, of course, with the Renew Zoo program, it's allowed me actually, interestingly, to branch out into amphibians. So that's why I say aquatic species, because we have 2 students now working on amphibians as well. One with the Renew Zoo program and one with another group partner that I have. So we focus not only on fish, but aquatic species in general. And today I'm hoping to tell you a little bit about what is reintroduction. A lot of people use the term loosely, but there are some very specific definitions. We'll talk a little bit, because I knew Dan was coming, about the history of reintroduction. I'm also fascinated by it. Some knowledge gaps, so I'll give you my thoughts on where we have real issues. And then finish off the day with talking about the really stellar work of some of the grad students from the Renew Zoo program that Albrecht led, and essentially tell you about how this grant is enabling them to do better work related to reintroduction biology. And of course today my goal, like the previous speakers, is not to drown you in data, but rather to tell you a little bit more about ideas we've had, things we've done, and if you want to read about the data, you can do that at your leisure. So we're all very aware in this room that anthropogenic-driven loss is a massive issue for biodiversity. My real interest in reintroduction biology came from literally the day I read this issue of Science. I don't often read it cover to cover, but this one particular issue, the cover got me, I have to tell you, that essentially this idea of vanishing fauna really piqued my interest, and there was a special issue talking about what are the problems, what are the benefits of biodiversity, and what can we possibly do about it. And this is in 2015 and 2013, they had 2 special issues come out in a row, and this issue really drove home to me the point that reintroduction biology is really a young idea. So how do we deal with these species or population extirpations? And of course we know that this Anthropocene defaunation, as they termed it, is not going away anytime soon. So the goal ultimately is to reverse this defaunation and do something about it in an active sense. We talked about this earlier with Andrea's work as well, that habitat restoration obviously is the key. Without a habitat to go to, reintroduction is essentially pointless. We know that we're trying to change some of this through policy initiatives. Some of it's successful, some of it not so much. But where we really are today, if you think about it, we're in a new era. This is really quite radical to think about. We're actually in what E.O. Wilson terms the era of restoration. And what we mean by that specifically is that we've moved away from a monitoring paradigm where we essentially catalog the fact that biodiversity is going down, 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 and we've moved away from that passive phase of monitoring to a more active phase of doing something about the biodiversity loss. And this biodiversity loss and some of its reclamation is under this area called restoration ecology. And within restoration ecology, there's a subject matter called conservation translocation, which is technically the umbrella term that reintroduction biology falls under. So if we look at the definition of conservation translocation, it's technically the deliberate and mediated movement of the organisms from any source, whether that be captive or wild, from one area to free release in another. And this is a nice review written by Swan in 2016, where she looked at the worldwide distribution of species, particularly marine and earth systems. She looked at coral reintroduction efforts, translocations. She also looked at all over the world these, and she was able to document this very clearly, that the number of cases of translocations or reintroductions, as you'll see, are becoming more and more common as a tool for conservation work. But if you think about the Translocation Continuum, which is often set out by Seddon and his colleagues, it really falls into three major categories, and you really have to delimit which category you're working on. So the first question you ask yourself is, are the animals of interest released back into their historical range? And for the most part, the answer is generally yes when it comes to conservation efforts, because when you say no, you have a much more controversial idea, which I'll talk about in a minute. So assuming the species of interest or the focal species in an area that used to exist, essentially what you're working on is essentially population restoration. And then you ask the question is, are the conspecifics currently present in that area? And if there are a substantive population, and we define this through PVAs and other statistics, there's enough individuals still in the area, we then say that this effort to restore them is actually technically reinforcement. And there's some examples like black stilts. However, if the species essentially is defunct or essentially not extinct, but essentially almost near extinction, we call it more of a reintroduction. That is to say, you're technically really getting individuals back to the area where they used to exist. And the Toronto Zoo, for example, works on Puerto Rican crested toads in terms of reintroduction, and of course the famous example of gray wolves. So the left-hand side of this continuum is quite standard practice, I'll say, for reintroduction biology. What's much more radical is the right-hand side, this idea of conservation translocation. That is to say, moving animals out of their historical range into a new range to save them. But often people have a big problem with this because essentially it's like creating an invasive species. And examples include certain bird species where essentially the last remnant of the individual population is on the mainland, but because of mammalian predators like rats, they're essentially going to get completely wiped out, so they move them to an island population where they've never existed. And this is very controversial. The other piece of this is also when range expansion is being contracted or changed because of climate change or different other human-induced factors, then moving the species outside of the range is very controversial as well. So the right-hand side is incredibly controversial, and I'm not trying to duck it today, but I'm going to focus more on the right-hand side, and specifically about this idea of reintroduction biology. As I mentioned, the idea of reintroduction biology is generally releasing a species back to its historical range with the ultimate goal of establishing a self-supporting population. So essentially what you want to do is you want to work yourself out of a job. You want to seed them out into the population, you want them to take hold into a habitat that's protected, and you eventually want them to be self-sustaining because you don't want to be continuously supporting them. So you want to work yourself out of a job eventually. And as I mentioned, if you think about it carefully, this is a radical idea because you're literally pushing back the bounds towards bringing the species back to its landscape. So you're having a drastic effect not only on the individual species, but the ecosystem it lives in as well. And there's a lot of debate about now, as we heard a previous speaker talk about, whether or not we should be preserving the ecosystem or focusing on specific species. And I think there's a strong work to be pushed towards the ecosystem perspective lately than it is species specific. But legislation is hard around that, as some of us know. So if you're not familiar with what it looks like, here's what it typically looks like in terms of reintroduction. On your y-axis, you have population size from small at the bottom to relatively large, and you've got some kind of time scale on the bottom. It ranges from either months, years, decades, or centuries, depending on the species you work at. So from reasonably recent to right now over on your right. And so what you can see here in this wild phase, essentially you have some kind of collapse of the species, so the population declines over time in some kind of function, and that function's slope often depends on whether it's human-induced over much longer periods of time, or is it happening because of a cataclysmic event. So then after some period of time, some individuals that are taken from the wild, so you end up having a wild foundling population in captivity, and you see this fluctuation here representing stochastic events. So whether it's an animal left outside that dies or a few individuals, these changes or fluctuations can be quite drastic because there's only a few individuals in the foundling population, and this has consequences also for the ultimate genetics. But ultimately you want to grow that population up in captivity, and eventually you need to manage that population. And if I walk you through some of the early steps, you see the decline, you see the population essentially being captively bred, and ultimately, like I said, you want to grow it to a reasonable size. And eventually, where it gets very challenging is when you have enough individuals to start seeding wild populations. The answer is, how do you manage that? And in fish, this becomes really problematic because you're producing sometimes hundreds of thousands or millions of fry or offspring to be sent out into the wild to do their job. So it becomes quite interesting to try to manage this population. And then over time, as you can see here, the idea is you want to select individuals for reintroduction, and oftentimes it takes multiple waves of introduction back to the wild. And your ultimate goal eventually is to work yourself out of a job because you hope that the species eventually starts accumulating in the wild and reproducing and eventually creating a self-sustaining population that you can essentially wipe your hands and walk away from them and say, success. Does that ever happen? Probably not. But the idea is this is what we visualize in terms of a framework for reintroduction biology. And of course, we know from many case studies this isn't the way it goes, but this is essentially, for your sake, sort of a mental template of what we're dealing with. So there's many examples from history. Dan being here, I had to show this. He actually mentioned this character. So William Hornaday essentially is very well known as some of the early work done on restoration efforts, reintroduction of the American bison. And unlike today where we satellite tag, we use really incredible technology with iPhones monitoring where they are right now on a live basis. Hornaday didn't do that. He actually had to send out physical letters, as you can see here, the American Bison Society. Hornaday sent out letters essentially asking questions like, please go outside and tell me, how many did you see today? How many males were there? How many born this year? And they would ask literal questions like that, and it would take months and months and months, if not years, to get that data. So a very different time, a very different way to communicate, and of course the attire is very different, as you can see as well. So gentlemen scholars were very important back then. As you can see that the times have changed radically since then. And of course we know that ultimately this led to eventually the United States providing this as an act, maybe one of the few acts that were finished in terms of making this one of the national mammals. So this is an example of something that's been going on for over a century, and it sort of points to the idea that restoration or reintroduction biology takes a long time and often multiple generations of people, if not the animals as well. So if you look at a few of the charismatic examples over long periods of history, I just sort of picked from the 1900s, from our example, the American bison. I didn't exhaustively list them, but you can see a variety of species that you might be familiar with, including the example of the peregrine falcons being reintroduced in New York, which is relatively successful. You've got a whole bunch of interesting dates. Here's the IUCN Reintroduction Specialist Group, formed not that long ago in relative terms. And I mentioned earlier that blew me away is that these first two textbooks in reintroduction biology only came about in the last couple of years, last 3 years. So the idea is that we see some successes, but of course reintroduction biology doesn't solve all problems. It's not a silver bullet. But what we can see is that there's some case studies that we can learn from. And one of the first case studies I worked on was actually the restoration of Atlantic salmon from Lake Ontario. And I'm going to sort of blast through this data for your benefit. But the idea is that we spent a lot of time for the last decade working on Atlantic salmon. And essentially in the early 1900s, Atlantic salmon were the most common top predator in Lake Ontario. Of course, now it's ironic. This is a picture that John Clayton took, one of our conservation authorities. And John Clayton demonstrated nicely with this picture that you've got the Pacific salmon that was introduced for sport fishing, which is technically invasive. And of course, you've got the Atlantic salmon weaning behind it because it's much smaller and struggles to keep up with Chinook salmon. But the idea was to restore a native species for fisheries purposes because it brings in lots of money and it's ecologically important. So as I mentioned, I've worked on this for about 10 years now with Lake Ontario. We knew from history that it's a highly abundant top predator. It's unknown from the Lake Ontario samples based on genetics whether it was landlocked, in other words, just stayed in the lake, or did it migrate out through the St. Lawrence. And for official records from based on CEEEC, which we heard about earlier, it was marked as extirpated in 1898. So we know it's been gone for a long time. The cause of it's very clear. It was obviously due to landlocked issues related to dams that were put up by European settlement. And of course, people harvest them for food as well. So the idea is that we had to deal with this in one way or the other. So we had a couple of options. There's different ways to deal with reintroduction at this scale. What you end up having to do ultimately is to sit down and pick source populations. When they're truly extirpated, there were literally no Atlantic salmon in Lake Ontario that they could ever find. You then go say, well, we're going to find other source populations to put back. And you've got a couple of strategies. You've got number one, which is to identify and introduce the closest surviving relatives in terms of genetics. So you go around scouring the areas of Atlantic salmon, looking for something suitable from a genetic similarity standpoint. That's one extreme of the continuum. The other end is this one here where you actually say, okay, I don't care about genetics. I don't say that very often, by the way. But if you say that and you say, I'm more interested in ecological suitability, do we have another sort of population that's living in an area that's similar to the lake you're working on? And of course, the last one, number three, is to introduce multiple strains with different traits, and we call this the experimental approach, so we can ask for a grant for it. And that's essentially what we did with Lake Ontario. We actually ended up stocking out 3 different populations because we actually had no clue which one to use. So we can look at some of the source population selections. These are a presumed best guess for Lake Ontario. We obviously had a freshwater life cycle. We're not sure about whether it was anadromous, whether it left or not. It definitely lived in the oligotrophic before the eutrophication came. And then genetic similarity, there was only 1 or 2 samples from the ROM that we actually genotyped, and that wasn't particularly conclusive. So what we did is we ended up having 3 source populations we knew we could get lots of individuals from, including Le Havre from Nova Scotia, Sebago from Maine in the US, and of course Lac Saint-Jean in Quebec. And we chose these because, as you can see from the little diagram here, they all reflect differences. One's more genetically similar, we assume, one's more ecologically similar, and one's kind of in the middle there. So this is that experimental approach where we stocked out 3 different strains into Lake Ontario to see which one might take. And this is really a learning process for us because at this scale, nobody had ever done this. And the scale I'm talking about is between our grants and the federal and provincial money, we spent tens of millions over about 10 years. So it's not trivial. The science coming out of it was very clear. NSERC funded, like to create some of this through SPG grants, and we led some of the grants in Western and other universities, and the province is part of it. All we need to tell you is that a lot of the students, in fact, some of your students, I wanted to put that up there, from Sudbury, who I recruited actually several of them, including Michaela and Chantel, who are undergraduates here, produced a large number of publications, probably about 65 to 70 publications in the 10-year span. And the idea is that we were very interested, the only point of these papers is to show you, we're very interested in the differences both ecologically and genetically between the strains to determine whether or not these strains gave us a hint as to which one's going to be more successful. And so what we did is not only did we look at the work in-house, we looked at it in the field, but we also did a kind of a summary study where what we did is we genetically tracked these 3 different strains in the wild. So essentially we did something called match plant introductions, meaning you just dump the fish in at the same time of the year from the 3 different source populations, and we stocked them as 3 different sizes: spring par, small fish, fall fingerlings, intermediate size, and 1-year-olds, significantly larger. And of course, each one comes at a different cost, and each one comes with different levels of domestication, for example. And then we genetically tracked them in the wild, so in Lake Ontario, and their returns. And we wanted to assess the optimal life stage and strain for both juvenile survival and ultimately adult returns, hoping that we could work ourselves out of a job and stop stocking them at some point or another. So what we use is dozens and dozens of microsatellites and other genetic techniques to look at the number of adult returns over time from 2007 to 2016, and we assess which population they came from. They either came from our 3 experimental crosses, so our populations, Lac Saint-Jean, Sebago, or Le Havre. The red ones are a little complicated because New York State also stocks some Sebagos, and they could have strays that came over. And of course, genetics is not perfect, despite what I tell some people, and so sometimes they're ambiguous. We couldn't tell what strain they're from. The take-home message is quite clear. If you look at the number of adults coming back to this is all the rivers in Ontario, you see there are very few for the number of fish. We stock out millions, we get dozens back. So I joke with people, this data point here is me. In 2007, I accidentally caught one of these fish, and I didn't actually believe it was one until the genetics told me it was. And this fish probably cost $250,000 to $300,000 because there was only one that year. So it's not trivial. And the idea is that you look at this pattern, and what you can take home from it is that Le Havre and Sebago, the red and blue ones, typically did much better. The Lac-Saint-Jean, the yellow, you don't really see any, as you can see. So that population really was not effective. So there was some clear-cut answers coming back to the management team based on our genetics. And the second piece of this was not just saying which population, but what life stage should you use. And we look at that, we can look at whether or not it's a yearling, 1-year-old, yellow fall fingerling, and spring par, and we have to adjust for differences in numbers. But the take-home message, essentially, if you look at the patterns, it's fairly clear without diving into the data that spring par, or that early release time, which has no or almost no domestication, ends up being the most effective. It's also the most cost-effective, which is also very convenient. So ultimately, what we're proud of is that we didn't just do the science, which is often what we do, and we forget about any implications of our science. We actually made sure that knowledge mobilization happened, and so we essentially came back and reported to our management teams and stakeholders and the public in general. We did a really big job about that, and we contributed to changing not just the practices in the culture centers or the hatcheries, but we also changed public perceptions on some of these issues. And of course, we focused the strategy on changing which stock life stage we did, and of course, we also had them drop the Lac-Saint-Jean stage, population, because it was completely ineffectual. So, we made big differences in a few years, and 10 years sounds like a long time, but in the grand scheme of things, it's certainly not that long. All right, so what I'm going to do is shift away from that case study, which got me initially interested in reintroduction, and tell you about some of my quick thoughts on what reintroduction could improve with and where we might be falling a little bit short. And then I'll come back to our Renew Zoo students and tell you how they're implementing some of these practices in their CREATE program that we have with Renew Zoo. So the first of these, the idea is to incorporate one of the things that Albert and I both do together, more together lately, but historically both follow this kind of line of thinking, is how can we incorporate ideas about ecology and evolution into these captive breeding programs or the zoo or aquarium programs. And simply put, I sort of sit back and I look at the Atlantic salmon, I look at other salmon I specifically focus on, and if you think about it, whether it's salmon or mammals, it doesn't really matter, but on all these mating systems, what's really fascinating is you look at these absolutely beautiful creatures, and what you notice very clearly in the wild is that they have polyandrous mating, meaning they mate with multiple mates. They have intense competition between both females and males. The mating competition doesn't just end with spawning. The sperm are actually competing amongst each other for the eggs. And fascinating enough, things like ovarian fluid around the eggs completely changes who wins out in that competition. So the female's not a passive participant. She completely biases who wins out. So there's a real tug of war. We have issues of mate choice where females are looking at these different males and saying, that is an amazing kite. I love that guy's kite. So the idea is that these translate into genetic quality issues. Life history changes, when they spawn is important, and of course the microenvironment, the river, the bed, all these things have radical effects on what genes pass on to the next generation. So you think about this, we spend oodles of time and many NSERC Discovery grants studying these kinds of questions, very basic science, and then all of a sudden we say, let's look at the captive breeding situation for reintroduction. And you start looking at it, and this is my most famous picture I like showing, because essentially what you've done in this one bucket, mixing sperm and eggs, or sperm technically known as milt in the fish world, is you've wiped out thousands of years of evolution in one swirl of that bucket. That's it, it's done. You essentially wiped out everything. There's no polyandry, because usually it's one-to-one mating. There's no choice, there's no competition, because there's just one bucket. The life history is excluded. They don't even ask where that male is from. They don't ask where the females are from in terms of when they spawn. The microenvironment in these stacks where they raise the eggs is neutral. There's no stones, there's no competition for oxygen. And of course, genetic quality, which outcome from both ecology and evolution, is completely absent. So you do these programs, you spend tens of millions of dollars doing this restoration for Atlantic salmon, and ultimately what you've done is wiped out thousands of years of evolution in one swirl of that bucket. So this bucket picture is essentially my entire discovery grant in one bucket. So the idea is I can just send this picture to NSERC and say, fund me for another 5 years. So the idea is we want to work this ecology and evolution knowledge that Canada is actually very famous for producing at a basic science level, and we want to take that basic knowledge and apply it. And so the idea is we want to take this with the Renew Zoo program eventually and do that. A couple other ideas. I get really frustrated with, I work a lot with the red-side dace, both in terms of biology and the field. This is a species that is listed as endangered, and it's going through changes with both the province and feds. And what I get very tired of is it takes them years. Policy sometimes takes years, decades, and essentially they're going to policy them to death, essentially, because by the time they actually get to something, this species is going to be snuffed in its infancy in Canada. And so what we did in my labs, I said I had enough of this. I went to Ohio, and we actually developed a cryopreservation. I thought I'd never have to do this in my life. I had to design a cryopreservation protocol with my postdoc to save genes from red-side dace. Why? Because in the Atlantic salmon, I learned that essentially we waited too long. We don't have any genetic samples from way back in the day to figure out what's different about the genetics. So what we do is we actually take the red-side dace, put them under anesthetic, flip them over, and gently squeeze them. And essentially they give us a sperm sample. We then freeze the sperm sample in liquid nitrogen through a process. And what that enables us is, I don't think about it as sperm, it allows us to save genes for the future. So this is what we end up having to do, because if we wait for Ontario to get its act together, the species will be gone. But now what we can do is they say, oh, we need a source population now to reintroduce. I wish we had some. I'm like, oh, we do. It's right there. Look, 519. I actually collected that one. So the idea is what we've started now, and I've just started to write another grant for this, is that we now just finished writing the first grant to establish the Canadian Fish Conservation CryoBanking Facility. And so this grant application going in now is for new equipment, and we have most of the equipment, but we'd like to get a big facility where you go around Canada and you preemptively, or proactively as I call it, collect genetic samples from species that are at a certain threat level, and by having those genes, if reintroduction is needed, you have a repository. And of course there's ethics behind this, and that's what makes the Renew Zoo program so fascinating, is that we have colleagues that are into both not just the culture aspects, not just the policy aspects, but also the ethical aspects of having a cryobank. So this is a really fascinating time, and this is the kind of grant that I'm really excited to kind of put together, because it gives us the ability to do something ahead of time. The third piece is obviously post-release monitoring. We spend tens of millions of dollars doing reintroduction, and for the fish world anyways, we do a crap job of tracking them. These are actually called bloaters. They have a very sad name because they call bloaters because when you pull them from the bottom of Lake Ontario, they essentially bloat because of the barometric pressure. And the idea is that we have to try to restore them for Lake Ontario because they're a native prey base, and these invasive alewives are causing problems. But the main point here is that we started saying, what's the point of stocking them out if we have no idea what happens to them? So we got a little bit better about using technology. These are VEMCO tags, acoustic tags. And what we do is we implant the smaller of the three tags into the body of the cavity of a subset of fish. We then take the fish from the hatchery. We actually have to take a truck, back the truck up to a tugboat, and the tugboat actually ferries out to the middle of Lake Ontario and then dumps the fish out into the lake, and we track them in real time. So these tags, actually, we have deployments of receivers, and it keep track of in the eastern basin of Lake Ontario of where these fish are going. The fourth point, and my last point for thought, is very interesting. I found I spent a lot of time with colleagues of mine. My old roommate is this guy named Julian Olden, and it talked a lot about Julian about invasive species. You'll hear about this in a second. And I hear a lot of ideas. I talk about reintroduction to him. He talks about invasive queen. I say, hey, Julian, reintroduction biology really has a lot to do with how many years you stock them and how many individuals. He goes, good point. How many times do you do it? What's the condition of the individuals? What are the biotic and abiotic factors? And ultimately, what you want is you want reintroduction success, and you walk away and you're done. He says, it's funny you say that. It sounds a lot like what I study. I study invasion biology, Julian says. And he says, essentially, what I study is how many invasion events are there? How many introduction events? I study how many propagules or individuals per event. I then study, interestingly enough, the condition of those propagules. I said, that's fascinating. And then, of course, the biotic and abiotic factors. So what we realized very quickly, him and I, having a coffee together, or many coffees actually, is that essentially we're studying the same coin, just different sides of the same coin. So what's interesting is there's a strong fundamental understanding for invasion biology. There's lots of modeling. There's lots of fundamental theorems. And what we can do is sort of kickstart the young field of reintroduction biology by stealing, essentially, ideas from invasion biology and saying, you know what, they're trying to get rid of them and essentially keep them from establishing, but they're starting to study how could they be successful. And essentially we have the opposite problem. We want them to be successful, but these same factors are driving some of those issues as well. So what him and I are doing is writing a review about how we can twine these two fields together and jumpstart invasion biology and reintroduction biology so that we don't have to reinvent the wheel. All right, so last few slides. Hopefully I'm on time. The idea is food for thought, of course. I told you these textbooks are relatively new, so there's a lot of work to be done. But you have to always ask the question, as previous speakers also mentioned, is, should we even reintroduce? There's a lot of issues around reintroduction. It's not a silver bullet. There are issues about cost. We talked about this word that we don't really understand, priority species. What does that mean? And the idea is, and we had a conversation with someone in the hallway. I can't remember if he's still here. Do we put a lot of money into 5 different species that have 0 chance, or do we kind of target a few? These are the kinds of issues that are beyond my scope of expertise, but there's questions we need to ask. But of course, the main point is that the conceptual
for reintroduction biology is very new. And what's fascinating, of course, is that the zoos and aquariums are being or getting involved in this program for a variety of reasons. And so reintroduction is not always possible for species like Atlantics because the footprint is way too big. But we know that zoos and aquariums, as we heard about earlier, have been part of programs like golden lion tamarins, that you got obviously the ferrets, the red-side dace at the Toronto Zoo, and obviously a bunch of amphibians. So they're getting into this reintroduction business, but they're going in somewhat blind. And through programs like the Renew Zoo program, we're trying to we don't have all the answers, they don't have all the answers, but we're trying to mesh what we both understand about natural processes. So a lot of times the zoos are really good about raising them, but they don't spend a lot of time in their native range, essentially outside the zoo. So we're trying to mesh those two fields. So I'll show you really quickly some of the students that are almost finished. So Marlena McCabe is a master's student who's essentially finishing this summer. Ashley just started. And of course, Andy Turco is a postdoc who will start from Guelph sometime this, I guess spring sometime. And the idea is just to give you a real snapshot of the power of this idea behind Renew Zoo that Albert came up with, which is integrating people in the zoo and aquarium business and people from the academic world and bring knowledge to the table from both ends of the distribution. And these examples hopefully give you a flavor of what we're doing. So I'll show you the first one. The first one is reintroduction of lake sturgeon to the Maumee River. And this is work done by the master student McKay, Marlena, I call them by their last name because I get too many of them after a while. And the idea is that the background to this essentially is that the Toledo Zoo is very keen to do this because the Maumee River runs right beside it. And what they do is they have this rearing facility made using a trailer, so you can see what the trailer looks like. And the idea is they want to pump water in from the river. They have good ideas behind why they want to do that, but they actually don't know whether it should be done or not, and that's what we came online to do. They're staffing the exhibit and the trailer with Toledo Zoo staff, 5 full-time aquarists, which is a significant investment, as you can tell. And the idea is that they want to rear them in either Maumee River water or municipal water. And the idea behind that has to do with this idea of imprinting. Do the juveniles that they eventually want to release into the wild as part of the reintroduction, do they need to have the smell of the Maumee River to come back to it eventually? And because they don't breed for 20 years, they wait this will sexually mature for 20 years, and they live up to 100 if you know. So they want to know, should we make sure they're on river water, or does municipal water work fine? And the idea is that what the student's doing, Marlena, is she's actually doing experiments where she spikes the water for half the brothers and sisters with an amino acid cocktail that smells like the river, and the other one she scrubs the water so it doesn't have the smell. And she's looking at behavioral trials of whether or not the juveniles want to come back to those areas. The other piece of this is they want to stock them into the Maumee, but they have no idea. They had no plan to follow out whether they did well. But what we talked about doing is obviously that acoustic telemetry I told you about earlier with the bloaters. And so the idea is Marlena, her second data chapter for her thesis, is focusing with the Toledo Zoo on the effects of those tags on the fish in terms of swimming performance, health, etc. And that's where the vets come in as well. So they're able to assess the health in different ways than we assess health or fitness. The other student I mentioned is Ashley Watt. She only started very recently, and Ashley actually fell into a windfall recently because she was interested in studying amphibians, and she's working on this species, the Mississippi gopher frog. And her focus is actually on the effects of senescence or aging, in looser terms, on gamete quality and offspring fitness. And if you think about it, a lot of these zoos that do reintroductions, they end up using very old captive stock because they're hard to come by, they're endangered obviously, or they're imperiled. So their stocks tend to get old, so they're breeding animals often that are maybe potentially much older than they should be, or older than they would be in the wild. And so the reason she hit a windfall recently is she started working with the Toronto Zoo, and then because we're in Windsor, Ontario, we're only 25 minutes away from the Detroit Zoo. So she got together between Toronto and Detroit, and all of a sudden with the staff at these two facilities, they said, we're doing a big march around all the zoos. So she gets to go to 9 different zoos this year because she's going to follow the different researchers that go from Toronto to Detroit to the Como in Minnesota to Dallas, and the list goes on. And she's going to collect data on the same project at all 9 places, at least 5 if not 9. And she'll collect very interesting data that will assess whether or not there's an impact of aging or senescence on fitness. And the way I'm going to skip that slide for the sake of brevity, but what she does, because I don't have my point, I can't do it from here, but what she does is she measures the sperm quality of the individuals, those are the males, young and old. She's got that clear paradigm. And then she does an egg extraction from the female, and then she does dry fertilization. These are actually videos, by the way, but I'll skip them for sake of brevity. And the idea is that she already did some of her internship at Toronto, so she can learn how to do these techniques. So when she goes to these other 8 or 9 zoos, she's not a newbie. So the Toronto Zoo is very useful at that. And then the other aspect is she wants to look at the downstream consequences for fitness or success. And so what ends up happening is when you fertilize these eggs, they take them to essentially the metamorph stage, and they physically put them in a box and mail them to Mississippi, where they get dumped into this one pond. So what she's going to do is what's called a split clutch design. She takes a female's eggs, she cuts it in half, takes half the eggs and fertilizes it with a young male, takes the other half of the eggs, fertilizes it with an old male, and because the eggs are from the same mom, you have no maternal differences in theory. And then what she does is she measures quality success in terms of embryogenesis, tadpole success in terms of swimming performance, and ultimately the metamorphs right before they go in a box, and off they go. So she's also going to have genetic samples taken from all these individuals, so we can genetically, like that Atlantic salmon I showed you earlier, we can genetically track the success of these individuals from whether they're from old or young dads in the wild. So taking it beyond the zoo facility and then taking them out into the wild. And it's that translation into the wild that I think brings our knowledge base to the zoo and contributes significantly to the project. And then the last person who hasn't even started yet, he's finishing his PhD at Guelph, is Andy Turco. And Andy Turco's focused on work with the Toronto Zoo, looking at red-side dace shown here. There's other species there, but red-side dace. And he's going to be doing translocation experiments and genetic tracking in the Rouge River, which is actually on the zoo property, so we have really good access to that property. And of course, he's going to do captive rearing on the zoo property with their facility. So he's going to have a public display, which we should probably talk to you about at some point. They had one at one point and kind of fell apart. And the idea is he wants to have this really strong public outreach component to really drive home the point that you can do something locally because the species got extirpated from the river in the zoo's facility. So it's kind of interesting in that sake. So I'll sort of wrap up and obviously acknowledge all the people I didn't mention by name, but you've got all these people from the Toronto Zoo, Ruth from Detroit, Kent Becker from Toledo, and all the other zoos that are going to participate down the road. Of course, we always want to be careful to mention the zoo technicians and staff because they are sort of the unsung heroes that do all this work on a daily basis. And we step in, do our research, and step out, but they're there day to day, 365 days a year. And I make sure my grad students don't forget that. And of course, none of this work would be possible without the Renew Zoo funding from the NSERC Create that Albert led, and of course the administration of it through TANIS as well. So with that, I'll take any questions for a couple of minutes, and then I'll pass the torch on to our next speaker. Yeah, so I'll take one or two questions. Yeah.
So I'm interested about the Cryo project and the idea that you're conserving genes for the future. I'm sure all the students know that genes are adapted to a certain environment. What if the environment is not there anymore? What are you going to do with all your genes?
Yep. So the question, about the cryo is kind of interesting. The Atlantic salmon is a good example of that. We've had the species extirpated for over 100 years. We don't know what Lake Ontario used to be like. We know it's very different now than it used to be. So that would be a mismatch for the cryo. The red-side dace is a little bit different as an example because their extirpation is imminent, frankly, within 5 to 7 years, we estimate. So that genetic variation probably is a better source population than having to go down to the southern US from a different designatable genetic unit. So we think the cryo is not the silver bullet, but under certain circumstances, like red-side dace, having something that's imminently extirpated is probably a good example where it'd be useful. But like you said, maybe for long-term, because we know the cryo, if you're not familiar with it, we've had examples in cod, for example, where they cryopreserve sperm for now 30 to 40 years and they're perfectly viable. So we could do long-term, but I think it's really meant to be done within a decade, frankly. Yeah. Any one last question before I pass on, or I'm happy to pass off at any point. And anyways, I'll be here afterwards if you want to ask anything else. So I'll pass at this point.
Thanks,
Yep.
Trevor. Thank you very much.